Peptides are stably bound to MHC molecules and also serve to stabilize the MHC molecule on the cell surface. An individual can be infected by a wide variety of different pathogens, the proteins of which will not generally have peptide sequences in common. If T cells are to be alerted to all possible infections, then the MHC molecules on each cell, both class 1 and class 2, must be able to bind stably to many different peptides. This behavior is quite distinct from that of other peptide binding receptors, such as those for peptide hormones, which usually bind only a single type of peptide. The crystal structures of peptide MHC complexes have helped to show how a single binding site can bind peptides with high affinity while retaining the ability to bind a wide variety of different peptides. An important feature of the binding of peptides to MHC molecules is that the peptide is bound as an integral part of the MHC molecule structure, and MHC molecules are unstable when peptides are not bound. The stability of peptide binding is important because otherwise peptide exchanges occurring at the cell surface would prevent peptide MHC complexes from being reliable indicators of infection or of uptake of specific antigen. As a result of this stability, when MHC molecules are purified from cells, their bound peptides co-purify with them, and this has enabled the peptides bound by specific MHC molecules to be analyzed. The peptides are released from the MHC molecules by denaturing the complex in acid and can then be purified and sequenced. Pure synthetic peptides can also be incorporated into previously empty MHC molecules and the structure of the complex determined, revealing details of the contacts between the MHC molecule and the peptide. From the sequences of peptides bound to specific MHC molecules, combined with structural analysis of the peptide MHC complex, a detailed picture of the binding interactions has been built up. And thus, peptides are stably bound to MHC molecules and also serve to stabilize the MHC molecule on the cell surface. Have a great day.